Okay, guys, there was a technical glitch, so we are back from the formation of the
Okay, guys, there
acetylcholine.
was a technical glitch, so we are
So,
back from the formation of the acetylcholine. There was a technical glitch.
so in the formation of the acetylcholine, what happens is that choline that is within the neuron, ECF, from the ECF plus acetate from the mitochondria and ATP and glucose both from the cytoplasm with the help of acetylcholine transferase, it forms acetyl-CoA. Now, this acetyl-CoA by endocytosis, okay, by endocytosis, which is endocytosis, which is a calcium-dependent protein, is released in the synaptic cleft. Now, this acetyl-CoA, okay, this by the process of exocytosis, it will be lysed out. It will be lysed out by exocytosis to the synaptic cleft. Okay, now from the synaptic cleft, this one it will come and it will bind on to the acetylcholine receptor in the postsynaptic membrane. Now here there is presence of acetylcholinesterase where in the synaptic membrane. So after the wave of depolarization is passed to the postsynaptic membrane. What is this postsynaptic membrane? This is the membrane of the skeletal muscle fiber. The acetylcholinesterase will break the acetyl-CoA and will be sent to the axon for reuse. Okay, now going to the diagram. Now, once this acetylcholine it binds onto this NM type of the acetylcholine receptor, immediately what it will do? Immediately it will it's a excitatory neurotransmitter, right? So immediately it will cause the influx of sodium into the sarcolemma. This is the sarcolemma. So it will immediately cause the release of the sodium and a wave of depolarization will pass through the sarcolemma. Now once this voltage gated sodium opens up and there is wave of depolarization in the postsynaptic membrane, it will open up the calcium channel, that is the dihydropyridine receptor. So this dihydropyridine receptor will cause release of the calcium in the postsynaptic membrane, and this in turn will cause the release of the calcium through the ryanodine receptor into the sarcoplasm. This is the sarcoplasm. So this is how the wave of depolarization, excuse me, initiates the action potential, initiates the contraction of the muscle. This is what is the event that is happening in the neuromuscular junction. So if I have to write the events in the neuromuscular junction, it will be stimulus via axon to the axon terminal, which is the presynaptic membrane. Okay, resulting in action potential in the presynaptic membrane, causing release of acetyl CoA in the synaptic cleft, then binding of acetyl CoA on the acetyl CoA receptor, right? Wave of depolarization generated on the postsynaptic membrane. Now, what are the use or what are the links of this presynaptic membrane and this postsynaptic membrane in various aspects? Now, the applied aspects of the neuromuscular junction. What is the applied aspect of the neuromuscular junction? Here, as discussed earlier also, botulinum toxin, it cleaves the snare protein. Various units of snare protein cleaves the various unit is cleaved by the botulinum toxin's various subtypes. So if botulinum toxin, when it cleaves the snare protein, the exocytosis of acetyl CoA is stopped. Okay, this is one. So, like that, this mechanism can also be used in the warfare, but we have studied it is like prohibited in the uses of the warfare. Second is that it is applied. It is used to check the insect and pest. How? Organophosphate poisons like malathion and parathion are used as insecticides. Okay, they inhibit the acetylcholine esterase. So once this acetylcholine esterase is inhibited, then the insect dies due to stopped contraction. So, in farming and agriculture, this neuromuscular junction targeted by this insecticide and pesticides is used to check and control the insects and the pest. Neuromuscular junction in pharma. Okay, what is the pharma link of it? Okay, certain muscle relaxants target the neuromuscular junctions. There are two types of muscle relaxants. Okay, one is the competitive and non-depolarizing. So here in competitive and non-depolarizing type of muscle relaxant, for example, tuberculin. Okay, so this is a type of a non-depolarizing type of muscle relaxant. How do they work? So logically, if you think non-depolarizing competitive means this particular drug, it is competing with the acetylcholine to bind on the acetylcholine receptor, obviously, right? So this competes with acetylcholine and stops acetylcholine binding on the acetylcholine receptor. So if this acetylcholine does not bind on acetylcholine receptor, there will be no depolarization. There will be no depolarization and the muscle will become relaxed. The muscle will be relaxed. How? Very quickly, let's look at the picture. Very quickly, let's look at the picture. So here you see, if acetylcholine is not binding on the acetylcholine receptor, so the wave of depolarization will not come from the presynaptic membrane to the postsynaptic membrane. So no depolarization, no contraction of muscle, but the muscle will stay in relaxed condition. This is one way of muscle relaxant. Another way Another way is the non-competitive and depolarization. Okay, this is the another method. So here what happens? Here, for example, succinylcholine. Now, succinylcholine, it acts just like acetylcholine and it has no esterase, means there are no succinylcholine esterase that can break the succinylcholine. So, what happens? The succinylcholine, it binds on to the acetylcholine receptor and induces continuous depolarization. So, the muscle will be in contracted state. Okay, so here actually it is acting, it is acting as acetylcholine, and it is like inducing muscle contraction. Okay, so these are the pharma link related to the neuromuscular junction. Certain toxins target the neuromuscular junction. Like, for example, tetrodotoxin. It blocks the sodium channel in presynaptic region. Okay, tetrodotoxin, it is from a fish that is called a puffer fish. Then we have the dendrotoxin. It is from a snake called the mamba snake. So we remember, like, it blocks the potassium channel on the presynaptic membrane. Okay. Another one, phonotoxin. It is from snail. That animal snail, that one. So it blocks the calcium channel on the presynaptic membrane. Okay. Now, going back to this picture, there are certain drugs that also act on the structures of the neuromuscular junction, like the drug salbutamol. It acts on the protein cluster. It kind of makes it unstable. The protein cluster that is present, that is supporting the acetylcholine receptor, becomes unstable. Here, this diaminopyridine blocks potassium channel. Okay, and drugs like pyridoxine blocks acetylcholine esterase. That means the digestion or the breakdown of this acetylcholine will not happen. So there are so many things happening in this neuromuscular junction. There are diseases that happens due to certain defect of the nerve neuromuscular junction. So two diseases we will discuss. Diseases of the neuromuscular junction, which is a part of our clinical link. Two diseases that we are going to talk about here It's like Anjum Nas. Hello. Hello to you also. So, two diseases that we are going to discuss here is the myasthenia gravis, and second one is that Lambert-Eaton myasthenia disease syndrome. Okay. Coming to the myasthenia gravis, MG. Myasthenia gravis, if you see, so very simply, in a very simple way, I'm just drawing it. This is our presynaptic membrane. This is our like postsynaptic membrane. In a simplified way, I'm just drawing for our understanding. And these are the NM receptors. Okay, so these are the NM receptors. This is our presynaptic membrane, and this is the excuse me, postsynaptic membrane. In case of myasthenia gravis, there are like antibodies. There are antibodies raised against this NM type of the receptor, NM type of the acetylcholine receptor. So this in myasthenia gravis, there are auto antibodies raised against the acetylcholine receptor on the postsynaptic membrane. So when there are antibodies raised against this NM receptor, what will happen? This receptor, this antibodies, the acetylcholine receptor will get destroyed. So as a result of which, what will happen? There will be not enough acetylcholine to bind on the acetylcholine receptor. So in context to that, one quick thing, just remember the there are about 60 synaptic vesicles that release acetyl CoA with every stimulus. But if the receptors are not there, if the receptors are less in number, even if acetylcholine is there, it has got no place to bind. So this will not induce action potential in the postsynaptic membrane. Now, this is one type of the myasthenia gravis in which the antibodies are raised against the acetylcholine receptor. Okay. In the second type of myasthenia gravis, antibodies are raised against the mask protein, against the MuSC, that is muscle specific protein. And we know MuSC protein, if we go back to the diagram, we will see this MuSC protein is one of the protein that is stabilizing the acetylcholine receptor. So here also, in absence of the MuSC protein, the acetylcholine receptor will not be able to bind, not be able to hold or get bound to the acetylcholine. The third type is third type of myasthenia gravis. Here the antibodies are raised against another cluster protein, that is low density lipoprotein 4. Okay. So what are the features of this? The features of myasthenia gravis as manifested in manifestation is that fatigue and weakening of the muscles, okay, which is called as the myasthenia fatigue. Then the reflexes are weak, but deep reflexes are preserved. Third is that the response to stimulus is decremental. The response to stimulus is decremental because there is lack of acetylcholine, because no adequate number of acetylcholine receptors, so acetylcholine cannot bind on the acetylcholine receptor. Okay, these are the signs. Now, what is the drugs used, or what are the drugs used? Drugs used are acetylcholine esterase inhibitors. Okay, glucocorticoids, cyclosporine, okay, cyclosporine, then rituximab. These are the drugs that are used to treat a patient who's suffering from myasthenia gravis, because if there is less inhibitor, like less inhibitor of the acetylcholine, if the esterase is like inhibited, then it will not break down the acetylcholine, because acetylcholine is very less in number due to destruction of the acetylcholine receptor. So the line of treatment is this one. The second disease of the neuromuscular junction is the LEMS, Lambert Eaton myasthenia. So this is actually, this is the disease of the, this is the This is the disease of the presynaptic membrane. Here, the PQ calcium channel, it is a type of the voltage gated calcium channel that is present in the presynaptic membrane, is destroyed. Okay, here it is destroyed. So, if it is destroyed, then without calcium channel, calcium cannot enter. So, exocytosis of calcium cannot happen. Cannot happen. Okay. Exocytosis of calcium will not happen. Features are, like as in manifestation, what is the feature that are found? Weakening of muscles, but incremental response to stimulus. And the person often shows doses of eyes, like the drooping down, the eyelids will be like drooping down. There will be like bilateral ptosis of the eye. Here, the line of treatment, the drugs used are mainly diaminopyridine. Diaminopyridine, it will block the potassium channels in the presynaptic membrane. So, efflux of potassium is reduced and repolarization not happen. So, as much as possible, the depolarization state is maintained so that there can be the induction of the release of the acetylcholine. Other drugs that are used, other drugs used are neothicmin, isostigmin group of drugs, which basically inhibits the breakdown of the acetylcholine, whatever is present in the synaptic cleft. So, this is our first part of the muscle physiology pertaining to the skeletal muscle fiber. In between, we had a internet issue, a glitch of the connectivity, but from after that, we finished understanding the neuromuscular junction in details. Tomorrow, we would again start with the remaining portion of the muscle physiology as a leaf 2. Thank you.